Hey, I'm Ben Kamens, the founder of Spring Science. We build AI technology to give scientists superpowers, and I'm going to take you through a very fast-moving demo here of our starting point, which is an AI-driven image analysis suite for high-content imaging data, running single-cell analysis at a very big scale, and generally pulling powerful information out of biological images. Our tools are used by big pharma groups, biotechs of all sizes, research groups, academics, and the main use cases are for novel compound and target discovery, elucidating complicated mechanisms of action and untangling those complicated mechanisms of action, running high throughput screens, comparing compounds to reference data sets, all sorts of things that help those in the R&D world drive their therapeutics programs forward. We are going to get in this demo into some quite advanced and powerful uses of Spring's AI technology and our foundational models to do things like automatically identifying and sorting cell populations, running self-service models to do so, automated quality control, detecting clusters in your data in an unbiased manner, so differentiating between healthy and diseased, or this drug and that drug, discovering novel interpretable phenotypes that are in your data that you might not have known are there, and in general supporting this very fast, highly optimized, at-scale analysis of large imaging data sets done at a single cell level. But in order to get there, we've got to work our way there, and so I'm going to take you through it. I'm going to build our way up through showing you how our technology enables that to be done at a high quality and trustworthy way. Now let's dive right into a demo. You're looking at a live instance of the actual application running on a real drug screen we ran related to inflammasome drug discovery. Everything is fast and optimized and searchable because it's all in our cloud. When you click into an experiment, the first thing you'll be exposed to is just fast navigation tools for exploring your imaging data. So no matter how many terabytes of data you're uploading and how many plates, you can quickly dive into specific wells, specific fields, view metadata at a plate level. If you're trying to get a feel for your experimental layout, you can flip back and forth between images and metadata, just fast navigation of all your data. At the same time, we do automated quality control analysis as soon as data gets uploaded. We've been generating imaging data and analyzing it with AI tools for 7+ years now, and so we've built quite a lot of techniques to establish whether data is AI ready or not, trying to knock out various experimental biases. We do row by row, column by column, edge effect searches, looking for any sort of disturbance or bias that may have come up in your batches, your different imagers that ran, things like that. And we've worked with all the imagers that are out there and many different protocol and experimental setups in order to build quality control systems and make sure that you are ready for AI analysis. So that's kind of it's table stakes. It's setting the scaffolding or the framework for the real power of what Spring can do, which is in these Analyze and Create tabs that I'm clicking back and forth on here. I'm going to start in this section here, and I'm going to call your attention to this Measurements dropdown. Something really important about the tools that Spring offers is we have two main paths, two main engines for driving analysis. The first engine is what we consider to be traditional featurization of these images. So all these microscopy images get uploaded to our system, and we will automatically do the generation of many traditional imaging features that folks may be used to from other approaches in the past. These are things like cell features, image level features, nuclei level features, cell area, compactness, eccentricity, things like that. Stuff that's based on intensity, texture, shape, segmentation of the cells. I'm going to dive into that right now, and then we're going to come back to the second form of analysis that Spring offers, which is purely AI-driven analysis, purely looking at each cell, extracting embeddings, and using those embeddings to drive very powerful workflows, many different workflows that give scientists new superpowers. Spring offers both, and both of them are set up automatically upon ingestion. You're not in there tweaking segmentation, tweaking settings, things like that. It happens automatically. When you choose something you're interested in, so maybe I'm interested in cell eccentricity, this again is one of those traditional features. I click on it, I get my experiment automatically broken down by, in this case, the drugs that have been screened defined, but I can break it down by what I want. I can subgroup by things if I'm interested in subgrouping by concentration for dose effect. I can, filter my data down to specific experimental conditions if I, if I want to filter down to specific metadata sets. You'll notice a lot of nice trust building things are in this application. Like when I start filtering my data, I'm not just showing the results, but I'm showing these little mini plate maps of which wells have been selected by my filter, so I can build my confidence that I'm selecting subsets of the wells that I like. Similarly, in the trust building domain, if I ever see a data point I'm interested in, whether it's one of these graphs or the QC graphs or anything that I see in the application, I can always just click on that raw data point and it will take me to the, so I'll click on the data point, it will take me to the raw image that generated that data point. And so here I can see these microscopy images that generated that data point I clicked on. I can overlay the numeric single cell values on top of every single cell that got calculated for, remember in this case I started by clicking on cell eccentricity, so we're looking at cell eccentricity values here, but I could select any feature that I'm interested in. So I can always click through the data to the raw images, and similarly in the lower right of the screen here, I can click through the data to a methods section and get a methods section quality breakdown of what produced the computational results on the screen, normalization approaches, computational approaches, things like that. And those things combined can really help you zoom through any result you see and get a real trust for the raw data and the approaches that generated the analysis. Similarly, there's a powerful tool here called image distribution, which I just clicked on. And I'm still in cell eccentricity mode because that's what I selected. When I go to image distribution here, I'm seeing a breakdown of all the image distribution values for cells across my entire experiment. And you can see there's these little vertical colored lines, and I can click at different points in the distribution. And I'm getting a sample of example single cell images, crops of single cells taken from points in that distribution. And so I can see what happens to the eccentricity of these cells as I go from the low value up towards the high value. And I can build confidence with my eyes that as this value increases or decreases, I understand what's changing in the biology of the cell. Now for this demo, that's very intuitive and fast to grasp when you think of cell eccentricity, but those of you in the cell biology space know that there's many complex things that you might want to measure which are subtle, and you really want to build your confidence that the values are changing, the images are changing in ways that you expect. And this tool really helps you build that trust as well. Okay, so I've raced through a bunch of stuff that's powered by what I was referring to as traditional image features. And again, all of this is happening automatically. We're extracting this for all of you. We've done this with many different cell types, and this is all a generalizable approach that saves you a ton of time. I'm now going to switch gears and start showing you some of the most powerful techniques that Spring Suite provides. And these are techniques that are enabled by our AI workflows and our foundation models that we've built to help power those workflows. And those models are powered by 7+ years of generating high quality single cell data to power this sort of work. Now, when you start this analysis, one of the workflows you start with here is this generation of these UMaps that are essentially laying out, I'm going to filter this down here for a second to a, this is a drug screen that we ran in the inflammasome space to try to find inflammasome inhibitors. What this UMAP is doing is it's laying out each well in a 2D space that represents all of the parts of single cell phenotypes that our embeddings think are interesting. And this is extremely powerful. You can color these wells by different compounds to see where different compounds are laying out. So here I've selected a specific compound and I can see where it's laying out in phenotypic space here. When I select a compound, then over here in the lower left, I can see which compounds are most similar or most dissimilar from it and what makes them most similar and dissimilar. If I lay it out by condition instead, so here you can see one of the interesting results from this drug screen, which was us clustering the phenotypes of primary human PBMCs that have been activated by this activator, ATP, those that have been activated by this activator, flagellin, and those that are inactive, the basal state PBMCs, what we would consider sort of healthy inactive state here in this DMSO vertical band. Now these phenotypic positions are entirely driven by deep learning analysis of the images themselves. So this is not applying AI to those traditional cell eccentricity features that I mentioned before. This is raw embeddings-based analysis taken from our foundation models applied at a single cell level to break down everything that's happening in the phenotypes of these wells. And this is very powerful because it lets you, in all sorts of contexts, produce results that you might not have otherwise found. So for example, here in this drug screen we ran, we're really interested in which of these blue wells are showing up in this orange band of phenotypic space, the DMSO-like band. And when I hover over those wells, I can see the rediscovery of some well-known inflammasome inhibitors, but if I were to keep hovering, I would then discover some novel inflammasome inhibitors, which was the point of this particular experiment. And in this case, it also helped us understand mechanistic action because we had different activators in the experiment and the like. This interface is very powerful if you want to just separate populations for any reason. It could be separating them based on different experimental states, like these inflammasome activation states. It could be wanting to understand healthy versus diseased phenotypes. Are those different, and how are they different? It could be wanting to separate responders and non-responders. All of these cases are cases that we've seen be very powerfully successful using these AI embeddings in our foundation models to help drive separation of phenotypes. We have extensive benchmarking work showing the outperformance of this in all sorts of standard industry settings compared to the traditional features, and that's something that we're happy to dive into and share case studies of for those who want to dive deeper beyond this demo. But I'm going to keep moving fast here because there's a lot to show. So this use case is one where you want to use the power of AI to better understand everything that might separate different imaging populations. Now, I'm going to take you through some use cases that start to answer the follow-up questions that typically come, which are, okay, I see that this population is different than this population, but what is different? Help me understand what's changing. Bring me to some mechanistic understanding here. One of those most powerful tools is this tool called PhenoSorter. And PhenoSorter is a technology that makes it possible for your scientists to do their own self-driven AI model building to quantify phenotypes they're interested in. So if I'm interested in, let's say, quantifying a phenotype that I think is relevant to my biology, in this case, ASC specs, I set up this model, and this system then starts showing me crops of single cells, and it's my job to start labeling them into different buckets I want to sort cells into. So here I'm going to label all of these as negative. I'm going to add a whole bunch. All of these are negative. Now, for this experiment, this demo, I just set up positive and negative buckets, but we've done this with many different buckets to quantify all sorts of different subtle phenotypes that anything your scientists can see, anything that they may be interested in, you can train your own AI model to quickly identify. Here, I'm looking for this kind of rarer phenotype. It has this pink ASC marker here. And I could just label these as positive. I'm going to ask the AI to help me, though. I'm going to click on Find Similar Cells. And it goes and finds cells that it thinks might be positive as well so that I can confirm it, label those as positive. And I can keep going here and keep labeling more cells. And this really doesn't take very long. I'm not going to take you through it for this demo. But once you get about 100 examples in each bucket and you click Train Model, this tends to perform extremely well. And we will hold out a test set, report to you the accuracy, let you refine the model, all sorts of advanced stuff. But the end result, the powerful result of this is that now that I've done that, in addition to those AI UMAP results, the graph I showed you that was breaking down populations, and in addition to these traditional cell image nuclei level features like area, compactness, eccentricity, now we have this new feature, this ASC positive thing that your scientist just trained very quickly, and you can see how that feature is changing across your entire experiment. And this is giving you the power of AI to quickly analyze things that typically take teams months of back and forth with computational teams, and computational teams are overwhelmed. This is a really powerful blocking and tackling tool and hypothesis testing tool for your scientists. Now, I'll also share a quick power feature here, which is in order to label these cells as ASC positive, to see these pink specks here, in this experiment, we had to apply a specific marker to this this palette. We had to add this ASC-specific marker to our imaging palette. Now, when you train the AI model to identify positive or negative ASC specs here, I could remove the ASC marker from that model and ask it to train again, and then see how performant it is. And what you are doing when you do this is you're asking, hey, can the AI identify this phenotype that I thought I needed a specific marker for? And in many cases, it is able to do this just using other stains, whether it's bright field or other typical cell painting-like stains like Hurst, concanavalin, phalloid, and things like that. And when you're able to do that, now you have freed up space in your imager to bring in another marker, build even more powerful biological palettes, or even just save money on reagents for a high throughput screen, that sort of thing. So some very powerful things that you can do here in addition to this. Now I'm going to keep moving. I just showed you now an AI use case where we wanted a very unbiased separation of different populations in your experiment. Then I showed you this tool, PhenoSorter, which is when you have a specific hypothesis or thing you want to measure, you can quickly apply your own AI model to measure it. Now I'm going to show you a tool called PhenoFinder. And what PhenoFinder is great for is a different use case where you want to discover unknown phenotypes that may be powering different results in your experiment. Now when I click on PhenoFinder, what you can see happening here is it's producing automatically. This is unsupervised, in AI terms, this is unsupervised single cell clustering. And it is producing these clusters here without me telling it anything about those clusters. And those clusters are defined by cells that the AI system thinks are similar to each other or dissimilar to each other. And so I'm getting all these different clusters of things it thinks are similar and dissimilar. And as a cell biologist, you might start to notice things that are interesting. You'll see that these cells are looking like they're starting to die in certain ways. And you can expand that cluster and see which phenotypes constitute this cluster or not. And maybe I'm really interested in cluster 20 here. And so if I'm interested in cluster 20, then on the right side, I can start to change which metadata is showing up as more or less overpopulated with respect to the existence of this phenotype. So here I can start to see that in condition ATP and in condition nigiricin, I'm seeing an overpopulation of cluster 20 compared to other clusters. And I can do the same thing for different compounds that are being screened or different donors or whatever it is that's most interesting to me. And this is very powerful for discovering novel biology that otherwise might have gone unnoticed, maybe because the cell clusters are rare or we just didn't know what to look for ahead of time. If you know what to look for, the previous PhenoSorter tool is great. If you don't know what to look for, this PhenoFinder tool for finding phenotypes is powerful. And we've seen this repeatedly surface novel biology that's very powerful. For example, in a pyroptosis-related line of work we were running, this technology surfaced for us the existence of a novel phenotype, which was these cells that had started to go through this pyroptosis form of cell death, this explosive, fiery form of cell death. And you could see the nuclei split apart, you could see pyroptosis start to happen, but then the cells would get stuck. So it's like pyroptosis started and then it got stuck. And we discovered this phenotype, and then that led us to certain compounds that were inhibiting the formation of pores on the outside of the cells. It was stopping the pyroptosis from happening. That led to the mechanistic understanding of those compounds and even led to clinical trials running for those compounds. So this type of technology can be very powerful for novel mechanistic discovery and novel biological understanding. Okay, so now we've shown a number of different tools that get powered by our AI technology. For the sake of demo speed, I'm not going to show you all of them. We have more. You can train your own classifier to quantify any metadata that you have in your experiment. So if you want to see if the AI can predict this experimental condition versus this experimental condition versus this, all sorts of stuff like that, you can use the supervised learner to do that. I do want to briefly show you this ranking tool, which is a use case that combines AI results and traditional features and lets you rank compounds in a screening scenario where you're trying to pick hits and the like. And when you do this, you might choose multiple features that you're interested in. So maybe I'm interested in some of those AI-driven positive and negative ASC features. Maybe I'm interested in different cell types that I've trained AI models around, like macrophages or clump lymphocytes. Maybe I'm interested in those that have a regular cytoskeleton, anything I've trained an AI model around. And I'll just pull in, all the nuclei features for the heck of it, just to show you if you're interested in a lot of features. Now, when you're using this tool, you can see what gets produced is this big map where every row on the left is a different drug that's been screened, and every column is a different feature, like ASC positivity or cell area or macrophages, whatever it is that I pulled in that I was interested in. And the heat map is showing me how, for each drug, each of those features is changing versus control. This is powerful if I want to start to map certain profiles to different control compounds and differentiate them. And importantly, I can also create my own scores. So as a scientist, I can go in and say, okay, I'm really interested in my custom score. I'm really interested in ranking my drugs in a way where I really don't want to see ASC positivity. And so maybe I'll give a negative weight to ASC positivity. And maybe I really want to see bigger, flatter cells for some reason. I give a positive weight to area shape area, and I can give a positive weight to this feature. And as I apply my human intuition and expertise to these features which are given to me by the power of AI. So this is combining human biological expertise with the ease of AI quantification. I'm re-ranking my drug results, as you can see on the right here, and then I can pick my cutoff point and call these my hits and choose the compounds that are ranking highest in this score. And so we see people using this in situations where you either want a very simple or a more complex way to analyze a drug screen and pick what's most interesting to you, powered by the features that AI gives you and that Spring's platform gives you. There's a lot more here. Thank you for putting up with me moving quite quickly through a lot of software. Our approach is to basically take the best and most powerful computational and AI tools available today, push them to their limit with high quality foundation models that we've built, and then put self-serve, nicely designed, fast tools in the hands of those R&D folks, in the hands of those scientists that are doing the most important work in the world, which is trying to find therapies for diseases. And we'd be happy to work with you and share more, dive into case studies and all sorts of stuff. Thank you very much.